Bienvenue dans votre émission Sciences et civilisations, علوم وحضارة. Nous allons voyager aujourd'hui dans le domaine de la biologie moléculaire et de la génomique. J'ai le grand plaisir de vous présenter mon invité aujourd'hui, Madame Zénib Rشييد. Je suis très heureux de votre présence. Nous allons faire le langage de la recherche, c'est-à-dire tous les langages de la recherche. Je vais parler en français, vous allez répondre en anglais et ce n'est pas du tout un problème.
Super, salam alaikum and thank you for having me.
Thank you. Alors, Madame Rشييد, vous avez fait d'abord ici vos études. Je dis ici le Maroc. Nous sommes ici au département qui est le vôtre maintenant de l'UM6P. Vous êtes dans le département de la biologie. Mais ici, je commençais par dire ici, c'est-à-dire le Maroc. Vous avez grandi au Maroc, vous avez fait d'abord l'université Aoula.
D'abord la FST, first the FST, Faculty of Science and Technology,
Oui.
in Stat.
In Stat, yes.
In Stat, and then I moved to Al Akhawain University
Yes.
To do a master's in biotechnology. My master's was actually between Al Akhawain University and the Albany Medical College in upstate New York. So I learned the fundamentals of biology in Morocco.
C'est ça.
But the love for the bench and, if you wish, the decision to become a scientist and pursue a career in science was taken at AMC. That's where I learned to love biology.
C'est ça. Donc, entre Aoula et l'État de New York, vous avez continué vos études, mais vous avez beaucoup voyagé, vous avez une mobilité de recherche très importante. Et dans mes émissions, je ne cesse de dire qu'il faut prendre conscience qu'un chercheur doit intégrer le paramètre de la mobilité, qui est très très important. Vous avez été en Suisse, à Berne?
Exactly, in Bern University. So after the New York Al Akhawain episode, I was accepted as a PhD student at KAUST University, King Abdullah University of Science and Technology in Saudi Arabia. This is a prestigious young university in which I learned all the omics and molecular tools. But my PhD was also done in collaboration again with the Vet Suisse in Bern University. And I learned basically the biology of the parasite, which is Teleria annulata. We'll talk about it in a minute, in Vet Suisse, and the omics technologies at KAUST University.
Alors effectivement, vous êtes spécialisé dans les technologies de omics, la génomique, qui n'est pas tout à fait la génétique. Vous allez faire vous allez donner la distinction dans un instant. Mais avant cela, avant de donner cette distinction importante entre ces deux domaines, j'aimerais qu'on continue votre itinéraire.
Absolument.
Donc, le Maroc, l'État de New York, la Suisse. À quel moment vous avez terminé votre doctorat? Dans quel lieu?
In KAUST University. So
Oui.
I finished my PhD in 2016, and then I moved to INSERM in Paris. I did my first postdoc at INSERM.
L'INSERM, c'est effectivement un institut de recherche médicale très important.
Exactly, at Cochin Institute.
Voilà.
That's where I did my first postdoc.
Postdoctorat.
Exactly, and then I did a second postdoc. So the first postdoc was mainly on the same microorganism that I studied for my PhD. I just deepened and compared my data to human data. And then for my second postdoc, I moved to LSTM, the Liverpool School of Tropical Medicine, where I studied another apicomplexan parasite, which is Plasmodium falciparum.
Donc, vous rejoignez, après Paris et l'INSERM, vous rejoignez Liverpool, l'école de médecine tropicale. Et finalement, votre domaine, c'est véritablement le domaine des parasites à ce moment-là. C'est ça que vous travaillez le plus.
Exactement. I work on pathogen genomics. So I try to understand the genome of parasites and the host cell interactions between the parasite and the host during the presence of the parasite. So I try to query or to question the host cell to understand how it responds to the presence of the parasite using omics technologies.
Voilà, et donc à partir de cette étude des parasites, vous ouvrez complètement le champ de la génomique. Alors c'est peut-être le moment, on l'a promis à nos téléspectatrices et nos téléspectateurs, de faire la différence entre les technologies omics, la génomique en général, et la génétique. Merci. À vous la pédagogie sur cette différence.
Thank you. We have to do pedagogy. We do teach students, right? So whenever we say omics, we add the suffix omics to any type of biological molecule. It means that we are taking a snapshot or an atlas of all those molecules within the cell at a time t, which means that if we add omics to transcriptome, it means that we are studying all the transcripts within the cell at a time t. Or if we say genome, for instance, we don't talk about all the genes, but we talk about the DNA, all the genetic makeup, which is the genes plus the intergenic regions. Same thing applies to phenome, like the phenotypes of the cells that are studied. If we talk about the metabolome, it means all the metabolites, proteome, etc. So this is a new area of research, omics. And not that new, but relatively new, which is the horizontal type of research compared to the vertical type of research. So if we say horizontal, it means that we are not targeting a specific gene or a specific transcript, and then we're not starting with a hypothesis. We're just comparing two different states.
C'est ça.
Okay?
La génétique se rapporte à un gène particulier,
exactly.
Alors que la génomique, c'est quelque chose
tous les gènes.
C'est tous les gènes, exactement, et donc une analyse beaucoup plus horizontale.
Tous les gènes et toutes les régions intergéniques, ça veut dire c'est tout le DNA de la cellule.
Oui, tout l'ADN de la cellule. Nous parlons de plus en plus aujourd'hui de séquençage du génome à haut débit. C'est une technique qui permet de détecter beaucoup plus de pathologies que par le passé, et même des pathologies que l'on ne cherchait pas et qui sont asymptomatiques. Et vous arrivez à faire ce séquençage du génome à haut débit ici. Ah, superbe. Et c'est quoi le domaine dans lequel vous vous orientez pour ce séquençage?
Well, what I do currently after I came back to Morocco, maybe I should say that I started forging my scientific identity as the independent researcher that I became. I'm no longer a postdoc. So I started to ask myself the question of what are the areas of research that will have the most impact in my country, in my society, and that I will focus on. So I decided to focus on the areas that are needy of attention from scientists, Moroccan scientists. And that's why I chose to work on the genomic surveillance. And now I'm gaining more and more interest into precision medicine. So for genomic surveillance, there is no better example than sequencing of COVID-19, of SARS-CoV-2. Okay, pandemic now gave us really an idea of how important sequencing the parasite is in order to be prepared for pandemics, first of all, by building the platforms and the systems for sequencing, but also to understand how mutations and genetic variants do relate and explain the dissemination, the virulence of the virus, and how we can use this data in order to adapt our treatments, our vaccines, and also our diagnostic tools like the PCR.
La surveillance génomique des pandémies est un point très important. Vous avez souligné l'enjeu de cette pandémie qui n'est pas complètement passée, mais on espère qu'on est au bout, la pandémie de la Covid. Et la surveillance génomique permet, vous avez dit, même de repérer parfois des mutations génétiques, c'est cela?
Exactly. This is exactly what we do. We try to when we sequence the genome, we find these mutations, but not only the mutations. We also detect insertions and deletions, what we call variants. Okay,
oui.
So what happens is that along with the sequencing, we also collect metadata. So metadata means data on data.
Yes.
All right. So we have the sequencing data, but we should also have data on the patients from where we got the samples, which means gender, age, symptoms, ethnicity sometimes, and geographic location, etc. It is this data that will help us make sense of the sequencing data. And then through this correlation, we can conclude that a specific mutation or a genetic variant has the ability to confirm a higher virulence for the virus, or a higher dissemination, or a lower dissemination, etc. It is through this analysis that starts from genome sequencing.
Et précisément, vous soulignez la collecte des données. Il est très important d'avoir une collecte des données. Vous avez dit ethnicity, c'est-à-dire des collectes des données suffisamment diverses à travers le monde pour qu'on n'ait pas toujours finalement une fabrication des traitements à partir de données collectées dans un seul endroit du monde. Pendant longtemps, on a fait de la collecte uniquement dans les pays dits riches, qui se trouvent être les pays anglo-saxons, les pays du nord de l'Europe. Et maintenant, précisément par cette nouvelle technique de génomique, il y a la possibilité de collecter des données partout à travers le monde parce que les maladies concernent le monde entier, ne concernent pas uniquement le Nord.
Yes, now you have raised a very important question about the democratization of the use of the novel technologies and access to this kind of data that can hugely impact positively the outcome of the disease, which is the prognosis, the health of the patient, and the whole access to healthcare systems in the whole world, not only in rich and advanced countries. Now, I'll give you a very good example about that. There is now what we call the polygenic risk scores. Polygenic risk scores are numbers that are calculated from different loci in the genome through sequencing, the human genome, and we try to stratify patients based on this collect of mutations, basically, say that this patient belongs to a group that has a high risk of developing a disease or a low risk of developing a disease. This is contrary to monogenic risk scores, which means that we do diagnostic based on one mutation, for example, or an insertion or deletion. So today, this depends hugely on sequencing, on population genomics, on sequencing large populations, and that means a lot of resources and know-how. Most of the population genomic studies done today come from patients of white Caucasian Europeans. And when we try to use these polygenic risk scores in order to predict the risk group of someone from another patient from another ethnicity, it doesn't work that well. And the numbers are nothing short of stunning. If you try to extrapolate those numbers to patients from other ethnicities, like Africans, they do perform 80% less than Europeans. Same thing goes with Asians. People from Southeast Asia would find that these scores perform 50% less than people from European ancestry. And that being said, we have to produce our own data in order to make benefits of the outcome of that data. So we can no longer be consumers of science. We have to be producers of science if we want to adapt it to our needs, the diseases that are most prevalent in our country, and to implement the findings of modern sciences and technologies in the healthcare system in Morocco.
Permettez-moi de reprendre sous trois formes ce que vous venez de dire et vous me direz comment vous réagissez par rapport à ma propre écoute de votre travail. D'abord, le monde africain est beaucoup plus touché par les maladies dites tropicales. Donc, il importe de collecter les données de ce monde-là plutôt que de traiter ces maladies à partir d'autres données. Cela tombe sous le sens. Mais en même temps, les technologies de génomique permettent de faire plusieurs sous-classes de population et et à partir de là, de cibler beaucoup mieux les traitements.
Exactly.
Et donc, en ayant les sous-classes de population, on ne va pas avoir un médicament générique pour toute la population compte tenu d'une maladie, mais on va pouvoir faire des traitements plus ciblés. C'est ce qui permet de parler de médecine personnalisée.
Exactement.
Dans un premier temps, quand on dit médecine personnalisée et ciblée, on pense que ça va exclure beaucoup de gens parce que c'est ciblé sur une partie des patients, mais au contraire, il y a la possibilité de l'avoir tout autrement en disant la médecine ciblée, la médecine personnalisée permet de faire des sous-groupes de population qui sont de plus en plus documentés par la génomique.
Exactement.
Est-ce que c'est à peu près cela?
This is completely true. Today, medicine has evolved, and the natural evolution of medicine does not rely on the onset of symptoms. Because historically, medical practice has relied on the treatment of the disease, starting from the onset of the symptoms and on the clinical signs that are shown by the patient. Today, medicine has evolved more towards a predictive medicine and into knowing how the patient is characterized compared to other people in the same population. Now, if we talk about the genome, this is one of the characteristics of the patient, or even the epigenome, now more and more used in the medical practice, that can give us more information on the patient and how he or she can respond to a specific treatment, and also how she or he are prone or at risk of developing a specific disease. So if we sum up now, medicine has evolved from being, proactive, from being reactive or curative, into being more proactive and predictive. And modern medicine today, we talk about the 4 Ps in medicine. Medicine has to be predictive, preventive, personalized, and also participatory.
Justement, sur cette médecine des 4 P, précision, prévention, participation et
personnalisation.
Et personnalisation, la prévention, certains disent elle n'est pas assez mise en avant, alors qu'on met beaucoup plus en avant la prédiction. Pensez-vous qu'il y a moins d'intérêt dans la médecine personnalisée pour la prévention que pour la prédiction? Elles doivent aller ensemble, prédiction et prévention, mais comment ces 2 paramètres jouent?
Exactly. We cannot prevent if we do not predict. And we have to predict the risk groups before preventing the onset of the disease using prophylactic treatments, for instance. A very good example is probably lung cancer, which I am more and more interested into studying, also for personal reasons, because someone that I hold very dear has lung cancer. In lung cancer, the main problem is the early diagnosis of the disease, which enables the window of surgery and localized radiotherapy. This is no longer possible if we wait until the onset of the symptoms. Now, another problem with the onset of the symptoms is that they are very similar to other respiratory syndromes. These are mainly cough and fatigue. So the patient himself does not recognize that these are symptoms of the lung cancer, and instead they wait for very long for these symptoms to fade away instead of visiting a doctor. So if we do not categorize the patients that are at high risk for developing lung cancer, for instance, and this can be very predictable, it can be very easy, male more than female, above a certain age, we have to study our population, epidemiology of lung cancer in our population in order to say, for instance, more than 50 or 55 years, also people who work in mines, people who do smoke. We can design some tests and early diagnostic tools that will be tailored and fine-tuned for our population in order to find the cancer, this type of cancer, early after its onset, and try to treat it with the minimally costly types of treatments, like we talk about surgery, we talk about radiotherapy. Now, if we move a little bit further, we will have to use chemotherapy, and now with the advent of the personalized treatments, also second line of treatment and the third line of treatment, which are very expensive and sometimes not affordable to all. So it is through the prediction that we can have access to the prevention.
Merci beaucoup, vous avez des qualités pédagogiques extraordinaires parce que vous m'avez fait comprendre que le lien entre la prévention et la précision, c'est la précocité, c'est-à-dire arriver le plus possible en amont. Vous avez cité l'exemple du cancer du poumon, et que plus on peut le détecter à l'avance, mieux il sera traité avec toutes les phases que vous avez données, mais surtout, ça permet de prévenir les évolutions péjoratives de ce cancer, et ça permet, par la prédiction, de pouvoir faire de la prévention, et tout cela signifie de la précocité, beaucoup de surveillance. On revient à votre domaine de recherche de prédilection, la surveillance précisément génomique. Je sais que vous vous êtes beaucoup intéressé à la malaria.
Oui.
Et que c'est une maladie qui touche beaucoup le continent africain, pour lequel on a essayé un premier vaccin, mais qui n'est pas si efficace. Il est recommandé depuis 2021 par l'Organisation mondiale de la santé, mais il ne dispense pas de pilules. Alors justement, vos recherches sur la malaria, elles ont été orientées, parce que bien sûr, les chercheurs ne travaillent pas sur tous les aspects d'une maladie, mais sur un aspect pour faire avancer l'ensemble. Votre recherche sur la malaria portait sur quoi exactement?
I did research on Plasmodium falciparum, which is the causal agent of cerebral malaria, and I tried to understand this interaction between the brain cells that come into contact with the malaria parasite and the parasite variants. Now, maybe I should explain a little bit about how these variants are different. When the malaria parasite infects a human being during the blood stage, it represents some antigens on top of the infected erythrocyte. These antigens will be recognized by the cells of the immune system in order to clear the infection. However, during its evolution, the parasite has found a way to evade from attack or clearance by the immune system by expressing different variants of the same antigen, so that it cannot always be recognized. These variants will differ in their length, in the modules that are presented in their order, and hence, because they are different in the length, they also differ in their cytoadherence. What do I mean by cytoadherence? These variants will cause red blood cells to cluster together and clog the microvasculature inside the brain. So what this causes is an obstruction of the blood flow, and also it will initiate some receptor-mediated signaling and downstream signal transduction pathways inside the brain cell. My role was to try to understand the involvement of these different variants in onsetting the cerebral malaria and how they do vary the pathogenicity of the cerebral malaria. So what I found is how we did that. We did that through co-culturing of the erythrocytes that are infected with different variants of the parasite, and the human brain cells. We put them in co-culture. And we could identify through transcriptomic sequencing, we could identify the pathways and the genes that are either activated after contact with the parasite or deactivated, or let's say downregulated after this close contact. We found the genes and the different signal transduction pathways, but what was very interesting is that among the pathways that were being actively silenced if the variant is more virulent is the prostacyclin. Now, prostacyclin is a known vasodilator. Okay, and it has been used, but very shyly previously to treat one case study only, also more or less in a few animal studies, but it did not receive the interest that it should have received, that it deserved for treating malaria. Now we have some evidence that prostacyclin is a vasodilator that can be a very good molecule for treating cerebral malaria. Later on, there were also some studies that did show that prostacyclin is also an effective anti-inflammatory agent and anti-apoptotic agent. Now, along with clogging the microvasculature in the brain, in the presence of the parasite, there is also what we call the loss of the brain-blood barrier. The BBB function is lost in the brain. It causes the swelling and coma, and it is fatal at the end. So now we have evidence that we can use prostacyclin as an efficient treatment molecule against cerebral malaria.
Mais c'est très intéressant. Là, c'est les atteintes cérébrales suite à une infection par la malaria. Ces atteintes cérébrales sont assez tardives. Quand une personne est atteinte, c'est bien cela. Ce ne sont pas des atteintes qui sont faites dès le départ.
Exactly.
Et c'est celles-ci qui sont malheureusement fatales parce qu'elles sont aussi liées à plusieurs handicaps. Et donc c'est sur ce moment tardif de l'infection que vous avez travaillé pour repérer ces cellules endommagées. Vous avez parlé depuis tout à l'heure et même au début de l'émission des différents variants. La génomique, comme la génétique d'ailleurs, parle de plus en plus de variants et pas de situations anormales. Je crois que c'est un élément éthique très important. Il y a avant tout à travailler sur les variants adaptatifs, des variants non adaptatifs. Mais quand on parle d'éléments qui ne sont pas normaux, automatiquement il y a une forme de discrimination qui est beaucoup plus forte. Donc, vous avez tout à fait adopté le langage éthique du variant et pas de abnormalities, de situations non normales. Aujourd'hui, dans la littérature scientifique qui est la vôtre et que vous parcourez et à laquelle vous participez, je tiens à dire aux téléspectatrices et aux téléspectateurs que vous avez beaucoup de publications. C'est le terme variant qui s'est imposé, c'est bien cela?
Exactly, exactly. Today it is a race. If we talk about epidemiology and about genomic surveillance, we talk about evolutionary processes. What that means is that the parasite, whether it is a bacterium or a virus or a protozoan, they are constantly evolving, especially for RNA-based viruses like SARS-CoV-2. They're continuously evolving, and through that evolution, they can be or become more adapted to our antiretroviral treatments, to our antibiotics, to our immune system, for instance. So we have to stay ahead of these pathogens in their evolution. And how that is done, it's through understanding the genome of those parasites. Now, imagine if we would not have had any sequencing technologies, if we wouldn't have sequenced COVID-19 at all, and we just notice that the pandemic is becoming more transmissible or more virulent, we would not have a clue. We would not understand how that happened. It is thanks to the omics technologies, to the study of those genomes, that we can attribute the phenotypes to their genotypes.
La génomique vous donne oversight, une vue d'ensemble, précisément dans votre autre domaine, la biologie moléculaire, et finalement, it fits together, ça va très bien ensemble, la biologie moléculaire et la génomique. Et vous veniez d'évoquer le système immunitaire, je sais que vous avez aussi beaucoup travaillé sur les cellules à macrophages. Alors, de quoi s'agit-il?
J'ai travaillé sur ça pendant mon doctorat. I worked on tuberculosis. I actually chose to work on tuberculosis because I had attended a seminar that talked about tuberculosis. So when I had to choose in the laboratory of pathogen genomics, I went straight away to tuberculosis because it fascinated me. This is an apicomplexan parasite that is unique in two aspects. The first one, which is fascinating, is that it hides inside cells of the immune system. It hides inside the cells that are supposed to attack and clear it. All right, so this is very fascinating. They have a evolutionarily developed mechanism to dissolve the parasitophorous vacuole and lie inside the host cell cytoplasm without being seen. Because it's through the parasitophorous vacuole that the cell of the immune system will recognize that there is a pathogen, that Theileria is inside, and it will try to attack it, destroy it using lysosomes. If the vacuole is no longer there, the host cell will not be able to find the parasite. So the parasite will be hidden inside the cell of the immune system, perfectly positioned in the cytoplasm to interact with the nucleus and with the signal transduction pathways to regulate the functions that are beneficial for the parasite itself. The second very interesting thing about Theileria is that it is the only eukaryote known to transform another eukaryote. All right? And this transformation is a cancer-like transformation. So the host cell becomes a cancerous cell. Characterized by all the hallmarks of cancer, including angiogenesis and controlled proliferation, growth without exogenous growth factor, migration and metastasis, etc., except for the genomic instability. So we knew that the parasite does not target the DNA of the host cell, so it had to target something else. My role was to understand the nature of these molecules that are targeted by the parasite and how they cause and they relate to this cancer-like transformation. So I did compare different kinds of infected and non-infected cells. This is comparative transcriptomics. I tried to make sense of the data of all the genes that go up, all the genes that go down, and through that I could recognize the genes that do play the highest role and the most important role in this transformation.
C'est absolument fascinant tous les domaines de recherche que vous soulevez. Ici, à l'UM6P, l'Université Mohamed VI Polytechnique, vous encadrez des thèses, vous encadrez des doctorants. Les tendances générales des recherches qui sont menées par vos étudiants sont lesquelles?
When I first came to Morocco, I started, as I said, writing my own projects and looking for funds, because this is what we do for a living. We write projects, we look for funds internally and externally, but I could not wait to see those projects funded, which currently they are in the genomic surveillance domain. But I had also to help colleagues, and I do work collaboratively a lot. I tried to help as much as I could using omics technologies and molecular biology tools to characterize the microbiome of the soil. And the students that I currently have 3 students that I co-supervise with colleagues that are microbiologists of the soil. And these students do studies that are related to biocontrol of fungi, to understanding the taxonomic and metagenomic variabilities and richness of soils from different agroecosystems in Morocco related to wheat and to maize, and also trying to understand the microbiome that is related to or connected to Fusarium oxysporum, which is a fungus that causes date palm dieback. It's the Bayud disease. So basically, we do not really have to be in our comfort zone to be able to produce. I was very happy to see that in the agricultural field, I also got a few publications, some in collaboration, some as first author. But I learned a lot from my colleagues, and I was happy to come out of my comfort zone.
Merci beaucoup, comme toujours, on apprend de nos étudiants et de nos collègues. Merci beaucoup. Je résumerai en disant que la surveillance génomique et les études génomiques sont suffisamment génériques pour pouvoir s'appliquer aussi bien à la recherche biomédicale qu'à l'agriculture. Merci beaucoup de votre présence parmi nous aujourd'hui. Je vous remercie, chères téléspectatrices. chers téléspectateurs, pour votre écoute attentive et je vous donne rendez-vous à une prochaine séance, Inch'Allah.